Our next speaker is Dr. Vijay Padal. Dr. Padal is a senior scientist specializing in cancer genomics, bioinformatics, and precision oncology. His work is in next generation sequencing and neoantigen peptide vaccines in advancing the field of cancer treatment. Dr. Padal, please go ahead.
Thank you, Dr. Shashanka, for that nice introduction. Hello, everyone. I'm Vijay Padul. I'm working as a scientist at Phoenix Life Sciences. My topic is genomics as a key enabler of personalized medicine. So everybody or almost all people want to live long and they want to live healthy and fulfilling life. So this longevity, this can be achieved in the background of good genetics. That means if the person is born with good genetics, then he will be susceptible to less number of diseases and will recover from diseases. And that will lead to a good long life. So what this longevity is dependent on, we are going to see the genetic components of it. So the longevity is also dependent upon the diet and lifestyle of an individual. It has a major role in how long a person will live. And various studies have identified there is a strong genetic component to longevity. So if you see biomarkers of aging, there are molecular, physiological, uh, and um, epigenetic uh, traits or markers that can provide an indication of how the person is aging. And though the genetic changes, they have been found to be changing throughout the lifespan of an individual. So there has been this quest of longevity with humans since long time, since the elixir of life. We have tried to live longer life, and in the modern times, due to the progress in the modern medicine, we have seen that there has been increase in the lifespan of the populations in different countries and world overall. So here we can see in the first table in Italy, we can say that in 1861, the average lifespan was 29 years, which has increased to over 80 years by 2011. This is a huge progress in achieving longevity, and that has been possible due to the modern medicine. We can also see in the second set of slides that in from 1990 till now, and there has been considerable increase in the longevity, and it will go on increasing up to 2030. Now, modern medicine has made this possible. We will see how the next step of modern medicine, that is personalized medicine, will help increase this lifespan more. So lifespan and health span, these are the two things that are needed to be considered. What do we want? We want increased lifespan, or do we want increased health span? I think everybody's aim should be increased health span. Nobody wants to be 90 or 100 years old and bedridden and taking medicines and not enjoying life. But if we increase the health span, that is the healthy lifespan of an individual, then we can do most of the increased lifespan that we have, that we can achieve. So there are definitions of aging and health span by Mokri. So aging, he defines it as the process of accumulation of consequences of life, such as molecular and cellular damage that leads to functional decline, chronic diseases, and ultimately mortality. Whereas health span, he defines it as the period of life prior to onset of chronic disease and disabilities of aging that is in good health. So what can we do to increase the health span? So the first thing we can do is to stay healthy. That means to not contract any diseases. And if we get any diseases, then we should recover from those diseases as quickly and as early as possible. So how can we prevent diseases to achieve longevity? So there are different hallmarks of aging and environmental and lifestyle factors that can affect those. So there are pulmonary dysfunctions that can cause due to the pollution, neurodegeneration due to the chemical contaminants in the food, type 2 diabetes, again a lifestyle disease, and there is a cancer. Cancer is a genetic disease which can have inherited component, and also there can be somatic components due to the exposure to carcinogens, which can be due to lifestyle of the individual. And cancer is also a debilitating disease, and it is also having an underlying genetic component. So to achieve longevity, one factor is that there should be disease prevention, and we would see how genomics can help in that particular um, area. So first, let's see what is personalized medicine, because our topic is personalized medicine. The key enabler of that is genomics. So we'll see to both of the areas, that is genomics and also personalized medicine. So personalized medicine is the next step of the modern medicine. The modern medicine currently, till now, it has a philosophy of one size fits all approach, where they prescribe the same medicine to all individuals with the same symptoms. So if you see in the diagram, this is a group of patients, and they have all genetic lifestyle variability, all kinds of variability is going on in the population. And here, if they have particular one symptom, then they will prescribe the same drug, and that drug will have different response in different patients. So it will have benefit to small subgroup, whereas other subgroup will have no benefit, and some other subgroups of people will have high rate of side effects. Whereas, what is the philosophy of personalized medicine is that there is a group of patients. We know that there is underlying genetic and lifestyle variability. Can we understand that particular individual? If that individual patient is there under the treatment consideration, can we understand that person's genetic background by creating genetic reports? Can we understand that person's lifestyle and basic constitution, health constitution? And depending upon that, can we prescribe a medicine that is more suitable to that particular patient's genetic constitution? Then in that case, each of these stratified group will get their specific treatment, and that will lead to better overall response to that particular drug in all the patients. So next part is genomics, as we are using genomics for this particular personalized medicine. So genomics, it is a study of genome architecture of an individual. That means the entire DNA genetic material of that individual. It can be sequenced and find out what are the different variants in those particular DNA, which are called as SNPs. And these SNPs, these generate different traits in an individual. So if a particular drug is provided, how long that particular drug will stay in that person's body depends on how fast that person's liver will process it. So underlying causes are the enzymes. And also, it also affects how that particular drug will bind to a particular protein, target protein. It also depends on the genetic architecture of that particular protein. So as it is different in different individuals, what genomics will do is that it will help us understand the genetic makeup of that individual, and based on that, we can tailor the specific treatments for that particular patient. So one example is inherited BRCA and APC germline gene variants that are present in population. So if a woman is found to be carrying a BRCA specific gene variant, which is associated with increased incidence of breast cancer or ovarian cancer, then that woman can take a preventive approach. She can undergo repeated testings, yearly testings, and also can undergo preventative lifestyles, and which can help into the prevention of breast cancer and also early detection, which can lead to proper recovery in the subsequent development of the disease. So this genetic test can also identify variety of different susceptibilities of certain genetic diseases that the person can develop and contract in the lifetime. So if we do genomic analysis of that person's genome, we can get that particular information and suggest a proper lifestyle changes to that particular individual, so that person can achieve longevity through prevention of diseases. So this is again an example of personalized cancer genomics. So cancer tissue, they arise due to somatic mutations, and some of these somatic mutations can be driver mutations. Here is an example of mutation that is BRAF V600E. Now there is a specific treatment available against this. That is, these are different antibodies, vimurafenib. If we provide this particular antibody to this person's treatment, it will inhibit this particular mutant protein, and the progression of the disease can be stopped. So this is one example of application of personalized cancer genomics. So in cancer, genomics is playing a huge role right now. It can enable identify what are the different driver mutations that the person harbors. Each individual person's cancer, though we can say that it is a particular type of cancer, it is different in different individuals, but the underlying genetic mechanisms can be different. And if we identify that, we can prescribe specific tailored treatment options to that particular patient, thereby increasing their chances of recovery. And in this particular aspect, cancer genomics is playing as a key enabler for this personalized medicine. So here, one of the latest example of the personalized medicine is personalized neoantigen peptide therapy. So as we have seen, cancer arises due to the somatic mutations, and these somatic mutations, they lead to the production of mutant proteins. And these mutant proteins can be recognized as an antigen by our immune system. So if we can, now that is possible in naturally, but in some cases it is not possible naturally, and therefore the cancer develops. But there is an option that we can exogenously train the immune system to identify those particular mutant proteins, and the immune system will be able to identify those proteins and attack the tumor cells, and therefore there will be a regression of the tumor and the cancer in the patient. Now, this has a lot of different genomics aspects, that is whole genome sequencing, RNA sequencing, exome sequencing, and also bioinformatic analysis to identify whether that particular antigen will be recognized by the immune system, whether there will be T cell activation, and after the activation, whether that T cell will go and attack the tumor cells or not. So all this process, major component of that is cancer genomics. So after that background, let's come to our research that our team at Genomics Life Sciences is doing. So this is a paper which is under review right now, for journal publication. So this is the overall outline of our research that we have performed. So there is cancer tissue, and there is a paired blood of that particular patient, and we do exome sequencing, RNA sequencing of those. There is an important component in the personalized neoantigen peptide therapy that that person's HLA background needs to be identified. And for that, the genomic analysis, we identify what are the HLA system that is present in that particular individual. Now, this our research aspect is that these HLA genes, which is an important tool to fight against the cancer in our immune system, is it getting affected by mutations or genomic changes or not? So here, there is a possibility that the HLA gene may get inactivated due to somatic mutation. It may get inactivated due to chromosome 6 loss, where this HLA system is present. And also there could be small deletions of those particular HLA genes, which may lead to the loss of the immune system's ability to fight the cancer cells. So here we have found in different number of samples. We studied 24 samples, and these are the different tumor samples in which we have identified there are mutations present. Most of these mutations were present in the intronic region, and one mutation was present in the exonic region. Though this mutation is found to be benign, but there is this result suggests that these HLA genes can also be target of mutations, and therefore this HLA system can get inactivated, adversely affecting the immune system to fight the cancer. Here are the 24 samples that we analyzed. Each of this HLA, and its result has been color coded, and we have studied it by using different softwares in order to increase the confidence in the HLA that we have deduced. Now, if you can see, in certain places, we have identified that those are indicated in the star. So if you can see here, in the normal, it is in a heterozygous state, but in the tumor, it is one copy has found to be deleted. So therefore, there is a loss of this particular HLA copy in this particular sample. So in some cases, all three are seem to be lost, but in some cases, only some individual HLAs out of HLA A, B, C are found to be lost. There is also possibility that there could be a chromosome 6 deletion. So as you can see in this particular case, chromosome 6 deletion is not there, but here in this case, chromosome 6 deletion is there. What does that mean? That one set of HLA A, B, C, and HLA class 2 HLAs are lost, and those are not available to fight the cancer in the personalized neoantigen peptide cancer therapy. So coming to the conclusion, as we have seen in through the background of the genomics and personalized cancer medicine, and also the research that we have performed, The genomic analysis, it can help to identify the longevity-associated genetic markers, whether the person will live longer or may contract diseases because of the presence of disease-associated variants, etc. And whether the person is suitable for personalized treatment options. That information can also be gained. And this cancer genomic analysis, it can help in patient stratification in cancer molecular subclasses, so that that particular subclass-specific treatment option can be provided to increase the efficacy of the treatment. And also it can help to identify personalized treatments that are suitable for the patient's genetic background. And in this way, genomics can help in the prevention of disease and recovery from the diseases, and therefore it can contribute to the longevity. Thank you for patiently listening.
Thank you. Thank you, Dr. Vijay. Thank you so much for sharing your insights from genomics for longevity. It's cool.
Thank you.
And